Welcome everyone to the next event of the Translational Neuroimaging Educational Program. We're really happy to have the speakers that are presenting today. I'm really excited. I'll introduce them in just a moment. But just for those of you who are tuning in for the first time, we wanted just to give a brief overview of the program itself. And in this program, we host a series of talks and presentations aiming to engage the neuroimaging and brain science community to enhance the translational application of computational neuroimaging techniques in promotion of human brain health. This educational program is nested within the Lifespan Working Group of the Enigma Consortium. In it, we host expert lectures and training workshops to promote the use of neuroimaging techniques to advance medical and scientific knowledge, and we welcome researchers at all levels and from diverse training backgrounds. The program itself is directed by Dr. Ruyang Gi at the University of British Columbia and myself at the Icahn School of Medicine at Mount Sinai, as well as the core advisory committee that you see here. For those of you who haven't already joined our mailing list, if you'd like to hear about upcoming events and news within the field, you can add your email to our mailing list and you'll receive updates of upcoming events. You can also follow us on YouTube, Twitter, Blue Sky, and LinkedIn, where we also post updates. All of the talks and workshops that we've included thus far have all been uploaded to our YouTube channel. You can scan this QR code if you like, which will take you there. And you can view or go back to any of the talks you might have missed or revisit any of them within this platform. And again, here are the QR codes to follow us on LinkedIn and Blue Sky. Twitter and Blue Sky. And for anyone who's interested in giving a talk within this platform, if you want to present your work and share some of the resources that you have developed for the translational neuroimaging community, we'd be happy to hear from you. Feel free to send an email to either Dr. Gi or myself, and we'd be happy to arrange something. For upcoming events on May 20th, we also have Dr. Ren, who is a principal investigator at the Changping Lab in Beijing, who will be presenting his work on deep prep, accelerated MRI pipelines and its applications. So that'll be May 20th. But for today, it is my pleasure to present Dr. Lindsay Oliver and Salim Mansour. Dr. Lindsay Oliver is a staff scientist in the Kimmel Lab with the Slate Center and the Brain Health Imaging Center at the Center for Addiction and Mental Health. She is also an assistant professor in the Department of Psychiatry at the University of Toronto. Her work aims to identify and target biomarkers of social cognitive deficits to inform and evaluate treatment options. Salim is a research methods specialist at CAMH's Kimmel Lab. With a software engineering background, he works primarily with neuroimaging pipeline processing and development for the functional diffusion and TMS data at the lab. He has been running and maintaining the BOONStim pipeline, which you'll hear more about, for 2 years now. We are very excited for both of you to present your work today on individualized TMS targeting in people with schizophrenia using Bayesian optimization of neurostimulation and presenting the BOONStim toolbox.
Thank you so much for the introduction and the invitation for us to present some of our work today. So I'll jump right in since you've kind of covered what we're going to be covering today, but essentially Salim and I are going to be outlining the Bayesian optimization of neurostimulation or BOONStim pipeline. So I'll provide some background information as well as an overview and some validation analyses. And then I'll move into some results using BOONStim to target social cognitive impairments in people with schizophrenia. And then Salim will provide a more applied portion of the workshop, covering how to run the BOONStim pipeline, as well as applying the outputs during TMS sessions. So repetitive transcranial magnetic stimulation, or RTMS, is generally safe and well tolerated, and it's been approved for the treatment of depression and substance use disorders. And current research supports its potential use in other disorders as well, such as schizophrenia. And RTMS involves non-invasively applying focal magnetic stimulation to the scalp, and this induces electrical stimulation in the underlying cortical tissue, and it can be targeted to specific brain regions. However, we know response rates to RTMS are variable, and they can be influenced by individual variability in both brain structure and function. So TMS localization and intensity are influenced by cortical geometry and tissue boundaries, and the positioning of the stimulation coil with respect to the brain, as shown here. And individuals also have unique functional connectivity patterns. So here we can see distances between personalized brain regions for two participants in purple and yellow, compared to a template region in black. And this work, led by our colleague Erin Dickey and others, has demonstrated stronger links between brain and behavior metrics using individualized functional magnetic resonance imaging, or fMRI approaches, versus group-based parcellations. So the majority of neurostimulation studies to date target a common anatomically based region across participants with the same coil positioning, and this may be suboptimal given this variability. So accordingly, there's evidence suggesting that closer proximity between the stimulated region and a post-hoc individualized target based on functional connectivity results in enhanced RTMS response. So here we can see greater improvement in depressive symptoms with a shorter distance from the optimal site. And individual variability in connectivity between specific brain regions has also been associated with the clinical efficacy of brain stimulation for depressive symptoms in depression, as well as negative symptoms in a small trial in people with schizophrenia, which is shown on the right here. And these findings highlight the potential for personalized connectivity mapping to identify brain stimulation targets. So there are openly available tools in both the fMRI and TMS spaces for people wanting to perform analyses in line with current best practices. However, available TMS optimization tools still require in-house software for fMRI data preprocessing and/or automation, and this may cause reproducibility and scalability issues. So our BOONStim pipeline was designed to fill this gap by providing an end-to-end scalable solution that integrates the current best surface-based fMRI data processing practices using fMRIPrep with TMS electric field modeling using SIMNIBs, or simulation of non-invasive brain stimulation, to identify an optimal TMS target and coil position. And it also uses a Bayesian optimization algorithm that allows for quick turnaround. So I'll get into some of the details a bit more here, but BOONStim, or Bayesian optimization of neurostimulation, was developed by a very talented former methods specialist in our lab, Jerry J. Chandra, and it's maintained by my co-presenter Salim. So it's fully automated from inputting MRI scans and specifying your target information to outputs that can be used in TMS treatment and experiments. So after inputting your T1 weighted and fMRI data in the required format, which Salim will cover more later, MRI preprocessing is done using fMRIPrep and the Ciftify toolbox, which was developed by our lab mate Erin Dickey, is used to project the data to the cortical surface. Then SIMNIBs and freeSurfer are used for T1 weighted head model reconstruction, or head model recon, which is also transformed to the cortical surface, so into a GIFTy format. And then the head model is registered to the fMRI surface using multimodal surface matching, or MSM-SULC. And users may define their own fMRI target workflows to derive regions of interest, or ROIs, and this can be based on brain activation or functional connectivity maps. Or you can adapt the default weight function workflow that we provide on our GitHub, or use an MRI coordinate. And I'll provide a bit more detail about this later, but after this, the target region is resampled to the SimNib surface to run simulations on. So if you're targeting a single coordinate, simple optimization mode is used to draw an ROI of a specified radius. And if a surface-based target is being provided, such as a functional connectivity map, weighted optimization mode is used. So BOONStim maps the user-defined target directly into the head model, in which the electric field simulations are then performed. And you can choose your optimization method, including Bayesian optimization or grid optimization, or the auxiliary dipole method can also be used, but only for a single MNI coordinate at this time. And Salim will talk more about the BOONStim output files, but they include optimized TMS coil coordinates and the Stokes correction factor to account for scalp to cortex distance, which are ready to use with TMS neuronavigation tools. So during optimization, electric field modeling is performed at different locations around the target to find the best TMS coil position to maximize the dosage over the target region. And unlike grid optimization, which tests every unique combination of coil angles and positions in a grid around the target, Bayesian optimization learns from tested parameter combinations to inform the next iteration. So this reduces dependence on the search space size and the computational cost involved. And BOONStim's Bayesian optimization process learns from a history of sampling points with basic assumptions about the smoothness of the underlying objective function, minimizing the number of iterations to find a global maximum, and it uses parallel sampling for faster convergence. So in terms of our validation analyses, BOONStim was run on 10 subjects on a data set intended for comparing preprocessing pipelines to compare our Bayesian optimization algorithm to the gold standard densely sampled grid optimization. So for Bayesian optimization, 30 optimizations with random starts were performed for each participant. And for grid optimization, this was performed using a 30 by 30 grid of locations with 36 orientations, resulting in more than 30,000 unique coil positions being evaluated. So the coil position with a maximal dosage or a simulated electric field stimulation over the target was selected as the optimal position for a given participant and ROI. So for both standard circular ROIs and functional connectivity-based targets, we calculated the percent difference between Bayesian optimization solutions from maxima detected using grid-based optimization and the number of iterations required to reach convergence. So BOONStim's Bayesian optimization was evaluated using standard ROIs of the dorsolateral prefrontal cortex, or DLPFC, which is the most commonly used TMS target, and functional connectivity maps of the dorsomedial prefrontal cortex, or DMPFC, which is a core node of the social cognitive mentalizing network, because BOONStim was developed as part of a clinical trial targeting this region, which I'll talk about more later. So our standard circular ROI targets were produced by drawing a circle of either 5, 10, or 20 millimeter radius around the selected DLPFC vertex. And for functional connectivity defined DMPFC targets, we took a meta-analytic map of the mentalizing network from Neuroscience and projected this onto the cortical surface. So that's shown here on the right. We then removed the DMPFC region and used a mask of the rest of the network. So here, this is the mask of the rest of the network without the DMPFC. So you can see the full network, and this is the rest of those regions. And we use this to compute mentalizing seed-based functional connectivity for each participant. So this is the seed-based functional connectivity map for one participant here. We then took a binarized DMPFC mask, and we applied this to this seed-based functional connectivity map. So we essentially overlaid the DMPFC mask onto this functional connectivity map to produce a weighted, personalized connectivity map of the DMPFC to the rest of the mentalizing network. And this is the region that we optimized over. So this is essentially the process that's outlined in the weight function workflow on our GitHub as well. So turning to the results, for both the standard ROI targets and the functional connectivity-based targets, Bayesian optimization converged to similar levels of total electric field stimulation compared to grid optimization across targets and participants in under 30 iterations. So this left panel shows the percent difference of Bayesian optimization solutions, where each line represents a different optimization. From the grid-based optimization maxima, which is denoted by the red dotted line, as the number of iterations increases for each participant. And I'm only showing the functional connectivity defined target results on this slide, but they were similar for the circular ROI-based results. And Bayesian optimization also converged within 5% error of the maxima detected by grid optimization, performing similarly again across standard ROI targets of different sizes and functional connectivity defined targets. So this right panel shows the percent difference of Bayesian optimization from grid-based optimization maxima, where each data point represents one of the 30 optimizations performed for each participant. And Bayesian optimization also converged in far fewer iterations than grid optimization, requiring less time. So here our plots show the number of iterations required for Bayesian optimization to reach convergence on the y-axis, where each data point represents one of 30 optimizations performed for each participant, with our standard ROI target results on the left and the functional connectivity defined targets on the right. And we can see that the number of iterations required for convergence ranged from 9 to 53 across targets and participants, compared to over 30,000 for grid optimization, with a runtime of about 16 minutes versus 120 hours with 32 allocated CPUs. So taken together, BOONStim allows researchers to perform automated, individualized TMS coil optimization with minimal setup, with the ultimate goal of enhancing the likelihood of response and improving clinical outcomes. It also incorporates a novel Bayesian optimization algorithm that solves TMS optimization over generalized ROIs, performing similarly to densely sampled grid optimization with both a low percentage error and substantially reduced runtime. And notably, its relative invariance to the search space size without sacrificing performance is significant in the context of clinical trials, where turnaround times between getting the fMRI data and delivering TMS treatment can be critical. So the three key advantages of BOONStim include its efficiency, its generality across target ROIs, and its integration of both established fMRI and TMS tools. So I'm going to switch gears now and talk about the application of BOONStim in the context of targeting social cognitive impairments in people with schizophrenia spectrum disorders, or SSDs. And individuals with SSDs often experience persistent and debilitating social cognitive deficits associated with functional outcomes, and these include impairments in both higher and lower level processes, for which there are currently no approved treatment options. So higher level mentalizing or theory of mind is thought to rely on a midline and lateral temporal mentalizing network, including the DMPFC, so that's shown in green here. And this involves complex mental state inference, such as intention and belief understanding, whereas lower level simulation or emotion processing is thought to rely on a frontoparietal and insular simulation network, shown in purple, and this includes emotion and action understanding. And in prior work, both DMPFC connectivity to the mentalizing network and mentalizing simulation network segregation have been associated with better social cognitive performance, suggesting that the DMPFC may be a target site to engage social cognitive circuitry. So based on this, this study aimed to use individually targeted 10 Hertz RTMS and intermittent theta burst stimulation, or ITBS, which is a more rapid and time-efficient RTMS paradigm, applied to the DMPFC to target the neural circuitry of social cognitive impairments in people with SSDs. So the goal here was really to demonstrate neural target engagement. And we aim to determine whether individualized DMPFC active versus sham stimulation would increase within mentalizing network connectivity and network segregation between the simulation and mentalizing networks from pre to post RTMS. So this data came from the neuromodulation of social cognitive circuitry in people with schizophrenia spectrum disorders, or MODSOCS study. And this was a randomized, double-blinded, sham-controlled feasibility trial. So participants were randomized to 10 Hertz RTMS, ITBS, or sham stimulation across 3 different sites, one of which was the Center for Addiction and Mental Health, where we are. And they underwent 10 treatment sessions, 5 days a week for 2 weeks, using a double-sided B70 active sham coil for blinding. And participants completed clinical and cognitive metrics, as well as an MRI scan, both pre and post-treatment. So the MRI scan included a structural T1 weighted scan, resting state fMRI, and the empathic accuracy task, which involves watching videos and rating how positive or negative they think someone else is feeling throughout the video. So of course, BOONStim was used to pre-process our participant data and identify individually optimized TMS coil positions. And more specifically, in the context of MODSOCS, participants' structural MRI and resting state fMRI data were input into BOONStim. And within the pipeline, individualized targets were determined based on personalized functional connectivity mapping to identify the region of the DMPFC most highly connected to the rest of the mentalizing network, as I previously described. Excuse me. And electric field modeling and the Bayesian optimization approach was then used to optimize coil positioning and maximally stimulate the individualized targets. So following pre-processing and modeling the empathic accuracy fMRI data, time series data were extracted from individually defined ROIs in the mentalizing network. So these included the dorsomedial prefrontal cortex and the left and right temporoparietal junction. And from the simulation network, so here we selected the premotor cortex into the inferior frontal cortex. And our outcome measures were change in within mentalizing network connectivity and between mentalizing simulation network connectivity. And we focused on effect sizes here to identify differences in connectivity changes between our active versus sham stimulation conditions, given the small sample size and that this was a feasibility study. So 49 participants started treatment and 46 completed treatment, and of those, 43 were included in our connectivity analyses after quality control. And participant characteristics are shown here for those who started treatment, and there were no significant differences in sex, age, or symptoms at baseline between the treatment groups. And turning to our results, neither sham nor 10 Hertz RTMS were associated with a change in mentalizing network connectivity. However, there was an increase in mentalizing network connectivity following ITBS when comparing the change in DMPFC to left temporoparietal junction connectivity after ITBS versus sham. So we were excitingly able to demonstrate target engagement here. We saw negligible to small effects in mentalizing simulation network connectivity changes when comparing active to sham stimulation. And importantly, in terms of safety, dropout rates did not differ across groups, and pain scores were low in all groups, suggesting good tolerability. So this feasibility trial demonstrated target engagement as well as safety and tolerability with ITBS to the DMPFC using our BOONStim pipeline. And the results suggest that ITBS may be effective in changing mentalizing network connectivity and potentially social cognitive performance in a larger clinical trial. So this highlights the utility of our BOONStim pipeline and individualized targeting approach. However, it should be noted that study limitations included the small sample size, the examination of specific node-to-node connections, and the limited ability to examine the potential influence of other factors, and a focus on effect sizes as a result. So I'll just briefly mention here that we wrote a review paper supporting a roadmap from moving from a behavioral target and translating functional connectivity biomarker discovery to individualized brain stimulation, which we've implemented in this work and ongoing work. And excitingly, the demonstration of neural target engagement in our smaller trial motivated a larger and longer efficacy trial funded by the Wellcome Trust that's currently underway to extend and confirm these findings, but using only ITBS to the DMPFC and our BOONStim pipeline. So here we're comparing the effects of individualized ITBS versus sham stimulation on social cognitive performance as the primary outcome measure. But we'll also be looking at changes in social cognitive network connectivity again. So the target N is larger here at 100, and the study is being conducted across the same 3 sites. And the trial also lasts 4 weeks instead of 2, including 20 sessions. And we're collecting data pre-treatment, post-treatment, as well as at 6 months follow-up. So this includes clinical cognitive as well as the same MRI data. So the results of this study could provide support for a novel therapeutic approach for social cognitive impairments in people with SSDs, as well as insights into functional mechanisms and longitudinal functional outcomes. So we're pretty excited about this. And I'll also mention that Salim is involved in a study led by Daphne Voyneskos and Colin Hawkow comparing standard TMS versus individualized targeting of the DLPFC in people with depression using the BOONStim pipeline. And this will be really critical to see if we're actually seeing better outcomes as a result of using individualized targeting and BOONStim in particular in terms of clinical outcomes. And with that, I will pass it on to Salim.
Thanks, Lindsay. Let me get my screen shared real quick. Great, if you guys can see that.
Yep.
It's all good? Okay, cool. All right, thank you all again for joining, and thanks, Lindsay, for going over the background and results for BOONStim. Just to go over and introduce myself real quickly, I'm Salim, and I work with running and maintaining the BOONStim pipeline for several studies, and I'll be going over some practical instructions now on how you can get BOONStim running yourself. I'll also end off with some notes on how you can apply the outputs from BOONStim directly to TMS sessions using the neuronavigation software Brainsight. So here's the overview of BOONStim. As mentioned before, BOONStim is a pipeline with stages including preprocessing, anatomical modeling, and Bayesian optimization that results in an individualized target for TMS targeting. I'll be going over the inputs, tools included, and outputs for the pipeline framework that puts it all together. Before we get started, in case you haven't seen this before, or just as a reminder, BOONStim is open source and all the code is available on our lab's GitHub at TigerLab, our github.com/TigerLab/BOONStim. I'll be going over a lot of this, but if you need to take a look at this at any point to look at the code templates and the documentation, it's all available there on GitHub. Specifically for the documentation, if you want to look a little deeper into how you can actually run BOONStim, the README goes into depth for the inputs and outputs and starting up BOONStim on your own. Just to kind of cement this, I will quickly pull it up. So here's the GitHub repo for BOONStim. And as you can see, the README is very extensive. It will go over how the code is organized, the requirements you need for installing it. I'll be going over all of this and how you can actually start running it as is. All right. All right, the first part of understanding BOONStim is your input data. Like Lindsay mentioned, we mainly need two things, the functional and anatomical data. Simply, functional data is used here for generating the correlation maps from the resting state scans to get a personalized target, and the anatomical scans are used for aligning the personalized target to the skull anatomy for the participant. How do we organize that data for BOONStim to ingest? For BOONStim, we use the brain imaging data structure, or BIDS, to organize our inputs. Sorry if you guys already know about BIDS, and I'm repeating myself, but it's a very cool and standardized way for organizing neuroimaging datasets. BIDS is important for reproducibility by ensuring consistency in data structure. It's easier for data sharing, and in this case, it's easier to automate quality control and preprocessing. I bring this up because BOONStim contains BIDS app pipelines and also is a BIDS app pipeline itself. Just as a kind of like brief overview of like how this looks, BIDS is organized in a hierarchical folder structure that is intuitive to how studies and datasets are typically collected. So as you can see here on the left, we have the raw DICOM data that's not organized at all. And on the right, we have a hierarchical approach for organizing this data. So as you can see, there's a root level for the dataset level information, like the dataset description and the participants.tsv. And then we have a folder for each participant and the dataset, and then by session and by modality. The key thing here to look at is the anatomical data, which is loaded under ANAT, and the functional data, which is organized under FUNC. If you're new to BIDS and you need your data to be converted, you can just rename it manually once you convert your data to Nifty, or use DCM to BIDS, which allows you to supply JSON describing what you want renamed from your DICOM files and how you want them to be placed in an output BIDS directory. And I recommend this approach because it's better for the long run. Now that we have our input data laid out cleanly, I want to go over how the software is packaged for you to use in BOONStim. As you can see, BOONStim relies on a lot of software to clean up the data, register to each other, and generate a target. These include the anatomical and functional preprocessing pipeline fMRIPrep, the surface connectivity pipeline ciftify, and the head mesh generation MRI image from SynNIPS. With all these dependencies, we need a reproducible, portable, and intuitive way to work with all the software. The containers are the answer to this. If you haven't seen containers, then the technical definition is that they are operating system virtualization, but literally think of it as a virtual container, literally as an object that holds stuff that you can take around anywhere you go. And this is very useful for importing software to multiple platforms. So here's how it works in BOONStim. We need all the software for BOONStim to run. For example, we need FSL, which is a suite of fMRI analysis tools. SynNIPS, which is, as I mentioned before, is an open source software for e-fields calculation and head models. Python, so we can run our Bayesian optimization scripts. NiPipe, which is a Python package for BIDS app pipelines such as fMRIPrep and ciftify. And these can all be compiled into a singular file called boonstim.sif, which can be read by Singularity or Docker. And then you can run this software on any system once you have this file. To repeat, like once you have this file and run on your system using Singularity or Docker, you can access any of the pipelines that we use for BOONStim, any of the software required. And this is kind of like a generalization of like how BOONStim is actually run with containers. We actually separate it out into multiple containers, so they're more lightweight and you can swap it out quickly. And these are all described in the BOONStim repo under the resources folder. As you see, this template params file.json. This is held in the BOONStim repo that you can pull at any time. And once you grab your Singularity or Docker images from Docker Hub, or you can build them yourself if you have a specific software version to use. You can just pull them to your system and then start running all the BOONStim dependencies as is. And yes, I'll refer to in the template JSON. And speaking of template files, the other thing I want to touch on is the additional scripts that were developed for aggregating all the preprocessed outputs I talked about and to generate a target at the end for treatment. Specifically, a lot of them are stored in the bin and modules folder in the repository, but I want to point out the weight function folder in the resources. These scripts are taken as a main parameter for BOONStim, so depending on what region you want to target, they can be changed here. We have provided templates using the DMPFC as an ROI, which we will use in a minute, but again, you can switch it out for any ROI that you want. Now, one more thing to discuss before we actually run BOONStim is how is this all put together? You can get this done by running a bunch of bash scripts probably and launch them all one by one, but we want a robust method of running all these tasks in a modular and scalable fashion. That's how we get to Nextflow. Nextflow is a language made for building scalable and reproducible data-driven workflows. Designed to work well with big datasets by scraping inputs or derivatives that you have, and it integrates with containers and Git for full control over your environment. All the pipelines and modules are recognized as separate processes, which opens a possibility for a lot of optimization, in which all the processes are running in parallel and being saved individually. This is really cool, and I'll show you how it works in practice in a sec. And yes. And if you want to get Nextflow installed on your system, you can run the main BOONStim function and the rest is taken care of for you. The other thing about Nextflow is I recommend it as a language as a whole. There's a lot of documentation for it and it works great. All you need is a Unix system and Java installed, and they have a quick start on their website at nextflow.io to get started working with it immediately. Now that we have everything, let's take a look on how to actually run the pipeline. So I have this command that actually runs BOONStim that's, again, in the GitHub repo, and it might look intimidating at first, but I'll go over each of the lines here. So the first 3 lines, we talk about, we are just calling the actual BOONStim main function called boonstim.nf, and that's already provided for you. It kind of lays out the overview of all the different processes that are run, and it refers to all the template files. The config file there also talks about what resources your system might have and all the different files that it might want to refer to as a the singularity images that I talked about before, and also the resources weight function config also supplies the weight function workflow that I talked about before. Then we have some inputs and outputs supplied by the bids and output directory parameters, and then we have a cache directory parameter which I didn't talk about before, but this basically, for all the different steps in BOONStim, it outlines it one by one, and each separate process is stored in its own folder that you can refer to later. So for example, if fMRIPrep is run, then you have a specific fMRIPrep folder for all the participants, and you can refer back to it. And the cool thing about Nextflow and BOONStim here is that if you have a step that's already run previously, Nextflow will look into the cache directory and then start running from there, so you don't have to run everything every single time. And then we have Bayesian. You can use Bayesian or record optimization here, but we're going to use Bayesian. And the subjects file for the bits directory that you have, and then the fMRIPrep and anatomical invocation files. I won't go over them too much, but basically they're just the parameter files for what modalities you want fMRIPrep to output, and if you want to refer to specific field maps, basically all the parameters that you can supply to fMRIPrep as is. All right, I'm going to quickly go to the terminal. Hopefully this text is big enough. I'm going to make it a bit bigger. But this is the BOONStim workshop folder that I made for you guys to kind of quickly show you how it's run. As you can see here, we have the bits directory, which is the main input folder that we need. And I grabbed a folder from a previous study that we worked with. And if you take a look at what's in the bits directory, as I mentioned before, there is the anatomical data organized in bits format with the T1 that we're going to be pulling from, diffusion data, which we won't be using here, and then the functional data with resonance scans to pull from, and that's how we'll be generating the connectivity maps. So now that since it's already organized as is, you can start running BOONStim with just that data. Now that it's organized, and I also pulled the BOONStim repo here, which all the files that I'll be using, and the BOONStim config repo that I have separately that you'll have to like also fill out yourself using the templates that we already have available. So with all that said, I'm just gonna, in the interest of time, I'm gonna just load up this command that I have here. And again, it looks exactly like the command that I had on the slide before, with calling BOONStim and all the inputs and outputs. So I'm going to show you how it actually is run. And I'm going to actually stop it for a second here so that I can show you the preamble of what Nextflow is outputting here. So when you run a Nextflow command, and specifically BOONStim, we print out all the different inputs and outputs and invocation files that are used under the hood so that you can, if you need to like switch out or debug something later, you can just refer to this and see exactly what's run. And this is good for like storing log files later. So yeah, once I run Nextflow, actually, you'll see that it's going to start running all these different parameters or all these different processes. And I can quickly go over the main ones. So the ones that it already finished is just grabbing the invocation files, and then it's actually starting to run fMRIPrep anatomical and fMRIPrep as is to get the functional data outputs. And then once those are run, it will automatically connect and run the later downstream pipelines. So MRI-TMS, ciftify, which all depend on the fMRIPrep outputs, will keep on going until you get the Bayesian optimization steps and the projection of the surface data onto the scalp, and all the way down until you get the QC outputs and the target. Now, I won't let this whole thing run because fMRIPrep and MRI-TMS takes a long time, and this whole pipeline end-to-end takes like 20 hours, since it does all the preprocessing and cleaning under the hood and optimization. So I'm going to cancel it as is, but before we get back to the presentation, I want to show you the cache directory. As I mentioned before, all of these folders are referring to the different processes that BOONStim runs. And as you can see, there's ciftify in here, and there's the run fMRIPrep folder here. And all these folders contain the different outputs step by step that BOONStim generates. And you can use this to maybe go back and debug in case you need to correct something, or if you need to fix a crashed pipeline, or you can just go back to check specific QC outputs, which I'll also go over in a sec. All right, now that I showed you kind of how to get started with the pipeline, I want to show you some outputs, since BOONStim has some specific outputs to itself and also the pipelines under the hood. So these outputs are very useful for quality control, which I'll go over in a sec as well. Basically checking that all the steps run correctly and that all of the steps, like the brain still looks like a brain and there's nothing that got broken in the middle there. And before I want to show you to kind of show you what the output folder looks like. If you look into this finished output, let's see. If you look at this finished output folder here, you'll see on the base level, there is the T1FS conform, which is the freeSurfer generated conform cleaned up process T1, which the TMS text will load directly into their neuronavigation software later. And then we have the results, which contains the actual neuronavigation coordinate that was generated by the Bayesian optimization in Brainsight.csv. And we also have the Stokes correction scale factors for both if you want to use the left side of the hemisphere or the right side of the hemisphere for treatment. And to kind of get a visual look, get back to the presentation. So the first thing that we usually check is that fMRIPrep ran correctly. Again, we want to check that the gray white matter segmentation, masking, and freeSurfer reconstruction worked well for all the anatomical and functional data. And we want to check that our surface-based analysis outputs from ciftify also look good. We want to check the fMRIPrep data that was generated was projected to the surface correctly, and that the EPI to T1 registration and seed correlation maps look sensible. In E-fields, which Lindsay mentioned is another TMS study that we're working on, we target the SGACC, but in the direction of the resulting E-fields generated by BOONStim. This means that we want to calculate the nearest gyres, but also in a direction that is parallel to the brain surface. So we have this output quality control file that shows the actual target location on the sulcal wall and the resulting target direction. And I'll show you, it's not just an image that gets generated, it's a actual 3D output. So you'll see that this is the actual target for the participant's treatment, and then the target direction is perpendicular to the gyrus in the resulting E-fields, and you can rotate it around to get a better look, but this is the mesh that's generated from MRI to mesh during the pipeline. And then the next thing, the TMS knob placement. Here we get the actual TMS knob placement for the TMS text to use later. And in this case, we're targeting the DMPFC, so we want to check that the line around the DMPFC region, or your target position that you use, is relatively perpendicular to the scalp, and that the line around the MT is roughly around the hand knob region and is also relatively perpendicular to the scalp. And this checks that the cortical distance and scalp projection measurements were calculated correctly. All right, to finalize all this, I wanted to go over how you can use these outputs for TMS targeting using Brainsight. As Lindsay mentioned before, we have a few studies investigating the efficacy of personalized targeting. In these studies, once a participant is enrolled and gets an MRI, it's up to our team to get a target using BOONStim and send it back over as soon as possible using the TMS text Brainsight suite for the participant's treatment start. And this is what they do with our target. To go a bit over the neuronavigation software that they use, Brainsight is a neuronavigation system developed by Rogue Research, who's based in Montreal, and it allows for real-time guidance of TMS coils in physical space using individualized MRI data in the software. Once you load in a T1, you can get a 3D visualization of the brain anatomy, and then we can get precise targeting of brain regions. This software is used as a standard in TMS studies globally. To start off with the Brainsight setup, yes, that's me in the chair. I visited the TMS text to get some boots on the ground research done for this part. But once the participant comes in for their treatment start, in this case me, the TMS techs start the Brainsight software, and they set the tracking camera pointing towards the participant's area while they're in the chair. They plug in the coil, which is used for treatment, and attach a head tracker onto the participant that the camera can sense. Once Brainsight is launched, you can load an MRI, and Brainsight will perform a 3D reconstruction from this T1 that we provide as its own project. To calibrate the participant's head model to the coil, the TMS techs calibrate the participant's head model using anatomical landmarks. So first they point to the bridge of the nose, they hit confirm, then the tip of the nose, and then hit confirm, and then both ears right above the tragus. Now the camera's stable. Let me run this video so you can see how it's actually used in practice. Treatment can begin now that the camera is stable. On the left here, there's a MagStim unit, which stores a lot of energy in capacitors. When a pulse is triggered from it, these capacitors rapidly discharge a high current through the coil. Depending on the E-field stimulation power that BOONStim also provides and the dose correction factor, the TMS tech directs the coil to the target provided and performs the TMS treatment for the participant. And here's a quick video of me receiving the treatment. It's kind of hard to see, but the coil is held still at the specified target that I provided on the Brainsight screen. And that's all provided by BOONStim. And that's it. Thank you all again for joining. As a quick overview, BOONStim provides personalized TMS targets using an ROI of your choice, and it has a focus on reproducibility, efficiency, and scalability through Nextflow. You can get started with BOONStim quickly today if you want to, through the templates and documentation available on the GitHub repo there. BOONStim outputs are easily used after running it by TMS targeting with Brainsight by the TMS techs on our side. And I want to thank the BOONStim collaborators and the research team that collected this data and administered TMS, our lab, and all the participants that joined for the several studies that BOONStim worked on. And also, Lindsay, do you want to quickly go over the postdoc?
Oh, sure. I also just wanted
Yeah.
to mention, yeah, thank you all for listening and the opportunity to present. And I wanted to mention that our lab, and specifically myself and a colleague, Nick Neufeld, are looking for a postdoc to work on some precision fMRI, social cognition, and digital biomarker data. So please scan the QR code if anyone is interested or if you want to share it with your group. And we'd be happy to take any